Liquid droplets formed from DNA display a peculiar response to enzymes. An international collaboration between Ludwig-Maximilians-Universitaet (LMU) in Munich and UCSB has now been able to explain the mechanisms behind bubble formation. A watched pot never boils. But researchers from Ludwig-Maximilians University (LMU, Munich) and University of California in Santa Barbara (UCSB) and found that's not the case when watching liquids formed from DNA. Recent advances in cellular biology have found that the molecular components of living cells, such as DNA and proteins, can bind each other and form liquid droplets that appear similar to oil droplets in shaken salad dressing. These cellular droplets interact with other components to carry out basic processes critical to life, yet little is known about how those interactions function. To gain insight into this fundamental process, the LMU-UCSB team used modern methods of nanotechnology to engineer a model system, a liquid droplet formed from particles of DNA, and watched those droplets as they interacted with a DNA-cleaving enzyme. Surprisingly, they found, in certain cases, addition of the enzyme would cause the DNA droplets to suddenly start bubbling, appearing just like boiling water. The bizarre thing about the bubbling DNA is that we didn't heat the system. It's as if a pot of water started boiling even though you forgot to turn on the stove, says Professor Omar Saleh from UCSB, co-leader of the project. However, the bubbling behavior didn't always occur. Sometimes adding the enzyme would cause the droplets to smoothly shrink away, and it was unclear why one response or the other would occur. To get to the bottom of this mystery, the team carried out a rigorous set of precision experiments quantifying the shrinking and bubbling behaviors. They found that there were two types of shrinking behavior, the first caused by enzymes cutting the DNA only on the droplet surface, and the second caused by enzymes penetrating inside the droplet. This observation was critical to unraveling the behavior, as it put it into our heads that the enzyme could start nibbling away at the droplets from the inside, notes co-leader Tim Liedl, professor at the LMU, where the experiments were conducted. By comparing the droplet response to the DNA particle design, the team cracked the case. They found that bubbling and penetration-based shrinking occurred together, and only happened when the DNA particles were only lightly bound together, whereas strongly bound DNA particles would keep the enzyme. Saleh notes, it's like trying to walk through a crowd. If the crowd is tightly holding hands, you wouldn't be able to get through. The bubbles, then, happen only in the lightly bound systems, when the enzyme can get through the crowded DNA particles to the interior of the droplet, and begin to. The chemical fragments created by the enzyme lead to an osmotic effect, where water is drawn in from the outside, causing a swelling phenomenon that produces the bubbles. The bubbles grow, reach the droplet surface, then release the fragments in a burp-like gaseous outburst. It is quite striking to watch, as the bubbles swell and pop over and over, says Liedl. The work demonstrates a complex relationship between the basic material properties of a biomolecular liquid, and its interactions with external components. The team believes the insight gained from studying the bubbling process will lead to both better models of living processes, and enhanced abilities to engineer liquid droplets for use as synthetic bioreactors. The research was enabled by an award to Professor Saleh from the Alexander von Humboldt Foundation, which enabled him to visit Munich and work directly with Tim Liedl on this project. These types of international collaborations are extremely productive, says Saleh. Thanks for watching. Do you like my video? Then show me your love by pressing like button. Please subscribe my channel and hit bell icon for new technology updates. Bye bye.